So in response to question 4, we observed elevated liver alanine transaminase, ALT, and aspartate transaminase, AST levels in our preclinical rat studies at the highest doses tested. High ALT and AST levels can be indicative of drug-induced liver damage that has severe implications for the safety of our drug candidate. However, high levels can also be transient or indicative of unrelated bodily stress. So we reviewed the recent literature's testing of new validated liver injury biomarkers that propose two further preclinical tests in rats to monitor the drug's potential to cause liver injury. First is 4-hydroxyphenylpyruvate dihydrogenase, HPD, an ELISA-detected liver tissue protein that serves as a biomarker for the hepatocellular necrosis at the micro and the macro scale. And 2, glutamate dehydrogenase, or GLDH, which is a blood plasma assay detected mitochondrial enzyme biomarker of hepatocellular necrosis with a prolonged persistence. If these biomarker results are negative, we will proceed with monitoring ALT and AST levels in phase 1 clinical trials by running standard liver enzyme tests, as well as testing for GLDH via an enzyme activity assay. So first, back to the basics to understand the problem at its core. ALT is a liver enzyme that converts the amino acid alanine to pyruvate, a metabolite of glucose, and the reverse transformation. It's an essential enzyme for the Cahill cycle, seen here, which links carbohydrate and amino acid metabolism. In essence, ALT helps convert amino acid building blocks to carbohydrate building blocks for energy use, or carbohydrates to amino acids for storage. ALT has a half-life of 48 hours and is specific to the liver. AST is a liver enzyme that converts the amino acid aspartate into oxaloacetate, a metabolite of glucose found in the Krebs cycle, and its reverse transformation. It's an essential enzyme for the malate aspartate shuttle, which moves glucose metabolites across the mitochondria to produce energy in the form of NADH, as seen here. NADH via oxidative phosphorylation is then converted into ATP, the primary source of energy a cell uses. AST has a 17-hour half-life and is located in a variety of other organs, including the heart, muscles, pancreas, kidney, and liver. So high ALT and AST levels found in the bloodstream are defined as the medical condition transaminitis. High levels are a trace or biomarker that liver cell damage or cirrhosis has occurred. However, given that these are traces of the damage, it's important to evaluate further to investigate the source of this. In addition, high ALT and AST tests have been known to give false positives in both clinical and preclinical tests. So elevated levels can be caused by drug-induced liver toxicity, which is a concern for our potential drug candidate. However, elevated levels can also be caused by a stress response. See, with high levels of a drug, as seen with this observed test on rats, may cause stress to the liver as it requires more work to metabolize, but not necessarily causing lasting damage to the liver. It can also be caused by a transient elevation record. We know the body expends energy in phases or bursts and recoveries, and these results may have been recorded at a high burst of liver activity. Thus, we don't know if the liver fully recovered or not. Or this could be an underlying condition like an autoimmune disease, ischemia, or viral hepatitis, which could induce liver damage unrelated to the drug we are interested in. In addition, AST can be found in numerous organs, as explained before, creating additional causes for transaminitis to be observed. This lack of clarity as to which cause we are working with begs further investigation into the source of these observed transaminitis. And in drug development, liver Histopathological necrosis and degeneration and serum liver enzyme elevations are most indicative of this drug-induced transaminitis uh, above the other types of liver damage. And this is what we aim to target with these additional preclinical and clinical tests. So we're reviewing a paper from the Safety Assessment and Lab Animal Resources at Merck. They tested the efficacy of 3 novel biomarkers in conjunction with and independent from ALT. The performance of each biomarker was assessed for its diagnostic ability to accurately detect liver injury. In 34 acute toxic rats, they analyzed for alpha glutathione S transferase, GSTA, arginine 1, ARG1, and 4-hydroxyphenylpyruvate dioxygenase, HBD, also known as serum F protein. They measured each biomarker's performance to accurately detect single-cell and extensive hepatocellular necrosis with ELISA technology assays taken from samples of the liver directly. So to test for the drug-related toxicity efficacy, thioacetamide was administered at 3 doses to result in hepatic necrosis. Animals were then sacrificed and tissue samples were analyzed. ALT and all biomarkers increased with each dose, but magnitude of change over control in lowest and highest doses was higher for the 3 biomarkers compared to ALT. These 3 biomarkers offer additional specificity in combination with ALT blood tests to detect for the drug-induced liver toxicity we are attempting to rule out. Though all biomarkers proved effective, HBD is the most of value because of its greatest specificity for liver injuries in both single cell and macro tissue levels of liver necrosis. In addition, TMPD, a muscle intoxicant, was introduced to rats to evaluate changes in the biomarkers. HBD was the only biomarker insensitive to muscle cell necrosis. This posits a superior detection of single cell necrosis in the liver specifically. HBD is therefore the optimal biomarker for early detection of liver damage. A different biomarker is advised for our second preclinical test. A 2007 research report by Safety Assessment Labs at Merck and Pfizer reviewed the enzymatic markers sorbitol dehydrogenase, SDH, glutamate dehydrogenase, GLDH, peroxonase, PON1, malate dehydrogenase, MDH, and purine nucleoside phosphorylase, PNP. They differ from the previous 3 biomarkers because they are all readily measured by serum photometric methods, which use commercially available reagents as opposed to the high-cost ELISA assays. The literature suggests that these markers, now examined collectively in this large quantitative study, provide additional information relative to serum ALT and AST tests. Of the 5 serum biomarkers, GLDH shows the most diagnostic value in comparison with ALT activity for our purposes. GLDH is more liver-specific over detecting muscle damage than the other enzymes in rats specifically. GLDH also shows advantage in detecting acute drug-induced liver damage in rats for 4 known hepatotoxic drug types, as seen along this x-axis of the figure, without inhibition or induction like the other biomarkers. This efficacy is of key interest to detect any candidate drug-induced liver injuries. GLDH also shows higher value increases and more prolonged persistence when compared to ALT tests, suggesting its higher sensitivity to acute liver injury detection. Both cross-species biomarkers are applicable to the rat species used previously, but may also be applied to other preclinical species if desired. They are also effective in predicting drug-induced liver injury, or DILI, in humans. Continuing with the same biomarkers has the added value for understanding the mechanism of action if the drug-induced liver injury is indeed observed. If these preclinical results indicate drug-induced liver damage, it's advised to reevaluate the drug's candidate safety. If not, the toxicology results of phase 1 clinical trials will better inform us of the overall safety for our drug candidate. So in 2019, Advances in Pharmacology reviews several accredited drug-induced liver injury or DILI biomarkers. Intrinsic DILI is due to the natural properties of a drug or metabolites and will be of primary importance for testing dosages safety in human clinical trials for our purpose. AST and ALT are not recommended on this list for drug-induced liver failure because these tests are nonspecific. After reviewing the list, GLDH, which was proposed for our preclinical study, can be easily analyzed with an enzyme activity assay in humans. GLDH is present in high levels as a result of cell apoptosis in the liver and will be the most effective for our purpose for detecting DILI. In recent clinical trials, GLDH has been elevated in humans in acute non-acetaminophen or APAP DILI cases. Most of these other biomarkers listed are more specific for APAP DILI, as it is the most common form of DILI experienced in humans. Also, GLDH shows efficacy in detecting DILI in patients with normal or low elevated ALT levels, adding to its predictive power, especially when taken in combination with ALT and AST levels. In addition, GLDH is more specific for liver injury, as shown in this image, than other tissue damage, serving an advantage over AST again. Though HPD is also found to be a predictive biomarker of liver necrosis in humans, it's less feasible than GLDH as a biomarker for the following two reasons. First, expenses of Western blot and tests are more expensive, and two, invasive procedures to collect liver tissues are required for that test. So in addition to the following three clinical tests, it is recommended that we choose a patient population with a diverse liver health statuses to understand how our candidate drug interacts with varying preclinical liver conditions. Liver health should be carefully and regularly monitored in all patients, and developing predefined safety measures in the case of drug-induced liver injury results is also essential when continuing on with clinical trials. Including these additional measures will ensure safety investigations into our target drug candidate. Thank you, and here are my sources.